All right, so let's talk about regulation of the cytoskeleton. So these are the two pages in your textbook, panel 16.3, page 905. This talks about the regulators of actin filaments, and then panel 16.4 on page 933 talks about the regulators of microtubules. I'm going to talk about them kind of together because a lot of them have a very similar function, right? These are things that you should recognize, even if you don't necessarily have the names memorized. The ones that we talk about are ones that you should recognize, so I guess you do need to memorize those names, but more importantly, memorize or understand what they do and what context they would be used. So in other words, how are they helpful for the regulation, and under what circumstances would the cell want to use that regulator in order to do its regulation. So what does it do, and why is that helpful to the cell, essentially? And so these are two good pages to study, but we're going to talk a lot about them here. All right, so the first thing I want you to think about is mixed group problem 4, actually. And so we'll have this as part of the Edpuzzle questions. So the concentration of actin in cells is nearly 100 times greater than the critical concentration observed for pure actin in a test tube. All right, so the critical concentration is the concentration whereby polymerization is automatic without any outside influence. So in other words, the critical concentration is the amount of monomers that you need in order to get polymerization. Right? So the concentration of actin in the cells is 100 times greater than what the cell actually needs, really, or at least what needs to be there in a test tube to get polymerization. Right? So the question is, what prevents the actin subunits in a cell from automatically polymerizing into filaments? So if we have that concentration that's 100 times greater than what's needed, at least in a test tube situation, what prevents them from just creating huge, long actin polymers that basically protrude throughout in a cell and burst through the membrane? So that's the first question. And then the second question is, why is it advantageous to the cell to maintain such a large pool of actin subunits in the cell? So think about those two questions and then go ahead and answer them as best as you can on the Edpuzzle. All right, so let's think about this together. Let's start with the second question there. Why is it advantageous to the cell to maintain such a large pool of actin subunits in the cell? Well, the answer to that is basically the whole premise of what we've been talking about in advanced cell all along, so that tying thread. The whole idea that post-translational modification is a much faster and more efficient way of regulating than regulation at the site of transcription and translation. So, in other words, by having a large pool of these monomers, you can deactivate them, you can sequester them away with inhibitors, and so then the effective concentration is an important term. That effective concentration is the concentration of monomers that are actually available to polymerize. So even if your actual concentration is 100 times greater than the critical concentration, if a lot of those are being inhibited at any given time, the effective concentration is closer to that. But they still are there. They are ready to be activated. You can release them, you can remove the inhibitors. By deactivating the inhibitor, you're actually activating the monomers, and so you can make the effective concentration higher almost immediately, very, very rapidly, by controlling the regulating elements. And you can even actually kind of hyperactivate the monomers so that they bind to the polymers much faster. So by having an excess amount of monomers, you can regulate the effective concentration by inhibiting them or activating them. And that's much more efficient than having to degrade a whole bunch of monomers when you don't want to polymerize as much actin, and then having to transcribe and translate and make a whole bunch of the proteins just when you want to make them. Instead, you activate and deactivate them much more rapidly and much more energetically efficient. Right? And so what are these things that regulate them? What prevents them? Well, for actin, we have thymosine. Thymosine is basically in the inhibitor. So when thymosine is bound to the monomers, they are unavailable to bind to the actin filament. So this is basically taking the actual concentration of monomers and making the effective concentration lower, because most of them are inhibited, they're sequestered away, they're not available to polymerize. On the other hand, profilin can promote actin subunit polymerization, so they're actually hyperactivating these monomers and making them much more efficient to bind to the polymer. So Free monomers are available to bind, but you can inhibit them with thymosin, and you can hyperactivate them with profilin. And the same is basically true with microtubules, although there really isn't a hyperactivator, so to speak. If you have free tubulin dimers, they are available to bind to a growing microtubule. But Stathmin, that basically is the inhibitor that binds to the tubulin dimers and prevents them from being able to assemble. So Stathmin is the microtubule version that changes the effective concentration and makes it lower. So again, that term, the effective concentration, is really important. That is the concentration of the monomers that are available to polymerize. And that's different than the actual concentration, which allows the cell to change the effective concentration quite rapidly because it doesn't have to wait for the monomers to be made and degraded and made and degraded. Instead, you just block them with thymosin or Stathmin, and then you inhibit thymosin or Stathmin, and thus releasing more of them to be available and increasing the effective concentration. Other regulation is nucleation. So what is the rate limiting step of polymerization? Well, the rate limiting step of polymerization is nucleation because you have to have a lot of monomers coming together at the same time. So the cell can actually promote that by having binding partners that basically nucleate this for them. So ARP23 is a complex that's already together, and it's set up perfectly for the actin filaments to bind to and then polymerize. And ARP23 binds to the minus end, thus allowing the plus end to grow, which we already know is the faster growing end anyway. ARP23 complex, as we'll see again, actually binds to an existing filament with a 70-degree angle, and that causes branching. So these filaments then branch, but all the plus ends are still out kind of pointed in the same direction. So keep this in mind because this will be really important for kind of the growth of that lamellipodia. Well, if nucleation of actin filaments is tough because you need a few actin monomers to collide correctly, nucleation of a microtubule is impossible. There are so many tubulin dimers that would have to collide at the perfect spot to nucleate a microtubule that it really just never happens. So microtubules have to be nucleated, and this is by the gamma-TERC, gamma-tubulin ring complex. So the gamma-tubulin ring complex provides basically a platform, a scaffold for these tubulin dimers to start binding. And again, just like the actin filaments, this binds to the minus end, which is why only the plus end of microtubules grow. You can have some actin filaments without ARP23 nucleating them. So there are actin filaments that can grow slightly at the minus end, albeit much, much slower, we know that. However, there are no microtubules that grow at the minus end because they are nucleated at the minus end and only grow towards the plus end. All right, so here's how we can accelerate nucleation. ARP23 for actin and gamma-TERC for microtubules. And so often these gamma-TERC molecules are at what's called a centrosome. And the centrosome tends to be this organizing area near the center of the cell where all the microtubules are going to migrate out from. And this is why we say that the microtubules, the minus ends tend to be towards the center of the cell where the centrosome is at, and then the plus ends radiate out towards the cell periphery. And the centrosome, if we have a cell that's moving, the centrosome is always organized on the side of the nucleus towards the direction that the cell is growing. And that actually allows for more of these microtubules to more easily grow towards the leading edge. And so we'll see in the primary literature article that this is a way to analyze and determine which direction a cell might be migrating. You look where the centrosome, where the gamma-TERC organizing center is compared to the nucleus. But they still can radiate outwards towards the other end. So the plus ends are out at the periphery, the minus ends are at the center where the centrosomes are. That's only true if a cell is not polar. So if you have polar cells, now we have cells that have two different ends. We have an apical end and a basal end. So my question for you is, why would we not want these microtubules organized at the center with all the plus ends radiating outward in a polar cell? Why would we want the minus ends at one side and the plus ends at the other? Well, the reason is we actually need to send things to different ends of the cell when we have a polar cell. The apical side is very different from the basal side. In these non-polar cells, especially ones that aren't migrating, the plus ends can just radiate out towards the edges of the cell just fine because all the edges are the same, right? There is no side that's different than the other. So basically everything just radiates out towards the cell periphery. And this means that things can migrate on microtubules by going on kinesins, which travel towards the plus end if they're going to go out towards the periphery of the cell, and by dyning towards the minus ends if they're meant to go the center of the cell. But if we're sending things to two different directions, now we need those north and southbound highways to be organized differently. This allows for things that are meant to go to the apical membrane to hitch a ride on dyning, which goes to the minus end, and things meant for the basal side to hitch a ride on kinesin, which goes towards the plus end. So now you can deliver your cargo to the two different sides because the microtubules end up organized in this way in the polarized cells. All right, next regulation is the speed and efficiency of polymerization. So if you can actually help actin monomers come bind to the filament, you can grow that actin filament much faster, and by growing it much faster, it actually is able to grow much further because you have much longer filament before hydrolysis starts to catch up. If you're growing it very fast, you can almost guarantee that the end is always going to be in the T-cap because you're growing it faster than hydrolysis catches up. So one cool protein that helps bring actin monomers to the plus ends of actin filaments is formin. This speeds up the elongation. And formin is this kind of cool protein. So in purple here, we have our actin monomers together in the filament. Formin is almost kind of this hula hoop type ring, and that hula hoop spins around the monomers as they're added so that it's always at the edge. And then it has these two long arms with multiple binding partners. All right, remember, profilin helps bind to actin and helps it grow faster. All right, it kind of hyperactivates it. So formin here takes its two arms, it's reaching out, it's always grabbing the next actin monomer, so it's grabbing them, and then it brings it here, and it basically puts it on the edge. And then it does a hula hoop around, and it's out on the edge again and grabs the next one. And so it's basically going like this all the while turning so that it's always at the end of the filament and grabbing and pulling monomers on there. And so that's how it can grow really, really rapidly. And this growth is really, really important. Right? So it's important for certain types. So do you think this growth here with formin would be for long, straight actin filaments or branched actin filaments like this? Well, because this is fast growing, you can get really elongated, straight actin filaments because they don't need to be branched. So this would be for things like filopodia and microvilli at the edge of cells where you need kind of long extensions. And so again, this is the microvilli here. Those would be helped by formins, kind of creating those long ones, and long, thin filopodia, those search engines with cells that are migrating, those are going to be created by formins as well. And that's because formin helps accelerate actin filament growth for long filaments. Then you can also regulate the filaments by the stability or instability of the filament or microtubule. So you can basically regulate how often that filament starts to fall apart. And you can cause it to fall apart, the cell can cause it to fall apart, or the cell can stabilize it and prevent it from falling apart. So one of these molecules that's really important is a molecule called cofilin. So in a normal actin filament double helix, it takes 74 nanometers to go one full turn. But if cofilin binds, that basically hyper twists it, and now it only takes 57 nanometers for this to go one full turn. And that hyper twisting basically is less energetically favorable. It basically now is kind of stressing it, and that can help it fall apart. So cofilin destabilizes actin filaments by hyper twisting them, and cofilin will cause them to fall apart. For microtubules, you can have MAPs. MAPs basically bind to the edge of microtubules, and they're going to stabilize them by binding alongside. So let's think about this for a second. We know that dynamic instability occurs when the hydrolysis catches up. So if this cap is no longer a T cap and now hydrolysis caught up and it's a D cap, that causes bending and basically peeling and collapse of that microtubule. But if you have MAPs there, you can stabilize the straight conformation, and now even if hydrolysis caught up, they are still straight. It's almost as if they were in the T form, even if they're in the D form. So when MAPs bind, these microtubules are not going to collapse and fall apart. However, if you want them to collapse and fall apart, even if they're in the T form, you can activate something called kinesin 13, and kinesin 13, when it binds to the filament, it basically causes it to bend, replicating what would happen in the D form. So if you want it stable, you bind in a straight conformation, replicating the T cap, which is stable. If you want it to collapse, you bind kinesin 13, which bends it and replicates the D form, causing the peeling of the microtubules. All right, so MAP stabilizes your microtubule, kinesin 13 destabilizes it. Another thing that you can do is actually stabilize it at the post-translational modification of these tubulin dimers. So I don't want to get into this too much, but this is actually important for the primary literature article. So these are post-translational modifications. So here's our alpha-beta dimer, and there are a few amino acids that kind of stick out along the side. They have this conformation almost like a tail out of the alpha and beta dimer. And so this C-terminus of the alpha tubulin can become detyrosinated. That basically means that the tyrosine is cleaved off. And when that happens, that post-translational modification allows these dimers to maintain the straight conformation. So this only happens when they are in the microtubule. When they are in the microtubule, this detyrosination basically can occur. That can get cut off. And now they are not going to undergo that bending collapse even if they are in the D form. So you can actually look for stabilized microtubules by staining for glue tubulin. Glue tubulin is that modification, the detyrosinated modification of this that represents straight, stable microtubules. All right, so you can also regulate the organization of the filaments, and this is particularly important at the leading edge of a migrating cell with the actin filaments. So you can create stress fibers. This is for attachment. So stress fibers basically allow the cell to attach in one place, and that stability elongates throughout the cell. And this is going to be more likely to be seen at places behind the leading edge where that cell is stable. So it's at places where it's contracting, it's places where it's stable, and stress fibers would also be more commonly found in a cell that's not moving. Right? On the other hand, we have filopodia and lamellipodia. So filopodia, as we saw with our foramen, these are going to be really tight parallel bundles of actin filaments that form these long protrusions. Those are the search parties, long protrusions, but they also often are dynamic, so they'll retract and another one will be sent out, and only the ones going towards the chemoattractants going in the right direction will be stabilized, and that'll guide the cell in that direction. But behind that, you need that broad lamellipodia front. So now we don't want long protrusions, we want the whole thing migrating forward together. And that's where you want that gel-like branched network. Right? So filopodia, the very leading edge, long, thin actin filaments. Lamellipodia, the broad edge kind of behind it where the cell's moving. That's a gel-like branched network of actin filaments, and then behind that are the actin stress fiber that allow for cell attachment and retraction. All right, so what molecules help organize those in the right way? We have three of them. We have fibrin, alpha-actinin, and filamin. So think about this for a second, what I just talked about. Which of these do you think would associate? Which of these regulatory molecules are going to help cause the long, thin, tightly packed filopodia? Which ones are going to cause the gel-like branching of the lamellipodia? And which ones might be kind of part of a stable aspect? See what you think. Well, actin filaments bound by fibrin. Fibrin is a much smaller molecule. That's going to bundle these together, right? And so this is a tight parallel bundle. On the other hand, alpha-actinin, this is associated with muscle fibers, particularly smooth muscle fibers like heart muscle cells and so forth, because the alpha-actinin sets these actin filaments perfectly distanced apart for the myosin bundles to come in and be able to bind and move those actin filaments. So alpha-actinin spreads the actin filaments out perfectly for muscles. Fibrin tightly packs them together for things like filopodia, microvilli in our intestines, and so forth. On the other hand, then filamin, all right, so there's the fibrin, filopodial extensions. Right? Filamin is going to help organize the branched networks. So here's the branched networks. This is the gel-like lamellipodia. Filamin forms the gel-like. Fibrin, filopodia, filamin, lamellipodia, alpha-actinin. Really, you might kind of talk about that with stress fibers. That it is there, but this is more of a muscle type of thing. All right, and again, just kind of showing that. Here's the tight bundling of long, thin filopodia. So formin would help the actin filaments become long, very long, and grow fast. And fibrin keeps them organized in this bundle for long extensions. On the other hand, that gel-like branched pattern is the lamellipodia at the leading edge of a cell. And then behind it, we have the retraction. So let's watch this in terms of cell migration. So here we have a migrating cell, and you can see that the lamellipodia just keeps moving forward. There's the main part of the cell body, and there's the retracting end. It's basically pulling itself forward. But there's the lamellipodia, that gel-like filaments are pushing forward. And if you were looking at this at high resolution with less time-lapse, you could actually see filopodia kind of leading the way, if you will, just at least to search. But there you can see the lamellipodia movement in the cell following it. All right, so now let's start to put some of this together to really talk about the regulation of cell movement. So remember that lamellipodia is kind of that broad face of the cell that's being pushed forward. And so that lamellipodia needs the branched actin filaments, and it actually needs branched treadmilling. In other words, it needs the minus ends to fall apart while the plus ends grow so that it's pushing that forward. And so if this is kind of the leading edge, the lamellipodia, and the lamellipodia is moving in this direction, we need to grow, so we need to orient the actin filaments so that the plus ends are branched but still all facing the leading edge, so that that edge will grow and the minus ends behind it will fall apart. And that is branched treadmilling. So treadmilling is when the minus end is shrinking while the plus end grows, and so the net is that the actin filament grows in one direction. Well, branched treadmilling is the same thing, but all of these branched ones are doing the same thing together. They're growing at the plus end, and that's pushing the lamellipodia forward, and they are depolymerizing at the minus ends. So polymerizing at the plus ends, depolymerizing at the minus ends. And that's branched treadmilling. So how do we make this work? Well, ARP23, remember, nucleates actin filaments. And so as we're growing, we want to nucleate new actin filaments rapidly at the leading edge. And ARP23 also nucleates them at these 70-degree angles. And in doing so, all of the plus ends end up facing the same direction, but kind of at these angles, 70-degree angles. So that gives us the branched treadmilling. Right? And then we can depolymerize behind it. So if you remember our cofilin, that is the regulator protein that hyper-twists the filaments and they basically destabilizes them. So we are nucleating at the leading edge, putting the plus ends forward, and we are destabilizing the minus ends behind with cofilin, and then that allows the monomers to be available to bind to the plus ends some more. They diffuse this direction and the whole thing's moving forward. So you need ARP23 nucleating at the very leading edge and cofilin destabilizing right behind that for that branched treadmilling to occur. And we can see that with staining. So red is actin, all right, and the green is ARP staining. So you can see that this cell would be migrating, well, let me ask you, which direction is this cell going? What do you think? Well, this cell is migrating upward or towards the top. And so the ARP is at the leading edge all around this lamellipodia because it's creating the branched actin format, and it's making those plus ends be towards the leading edge. And so there's still actin there, but it's basically just overshadowed by the green staining with all of the ARP, creating branched treadmilling with the plus ends towards the leading edge. Right? And so if we take a look at this a little bit more closely, you can see here now, which direction is this cell moving? Well, this cell is moving to the right, right? Because you can hopefully start to see the shape, the lamellipodia, and the leading edge moving forward and the retracting edge pulling up. So now we've stained with cofillin in green, red is still actin. So now you can see at the very leading edge you have actin, right? And ARP would be there too if we had stained this for ARP, but cofillin is right behind that. Cofillin is destabilizing these branched actin filaments and therefore allowing treadmilling to occur. We are activating nucleation, plus ends forward at the leading edge, destabilizing right behind it, and you're going to get active treadmilling, pushing, in this case, the cell to the right. And so by working together and being organized in this way, that cell is moving in one direction. You have ARP nucleating the plus ends forward, and right behind that cofillin destabilizing the minus ends, branched treadmilling. And so this is part of that cell movement. In order for the cell to move, you need three aspects. One is the protrusion, and that's what we were just talking about there. The protrusion is pushing that leading edge forward, and that's accomplished by the branched treadmilling, right? With ARP nucleating positive ends forward, cofillin destabilizing behind. And then, as it protrudes forward, now you need to create new attachments, because if you didn't attach, there's nothing for this to use to pull the rest forward. So you get protrusion with actin polymerization, branch treadmilling. Then you get attachment with cell junctions, some focal contacts. And then ultimately you have to have retraction, often referred to as traction as well, contraction of the rear. So the rear has to be pulled forward. And we'll see that here in a second. So how is that rear pulled forward? Well, now hopefully you can see that this cell is moving towards the top. Right? Now actin is in blue, so you can see the actin filaments and lots of actin at the leading edge. But red is myosin, and remember, we haven't talked about it yet, but myosin is that motor protein that pulls actin filaments. Okay? And so it's going to help pull the back edge forward. So let's think about how this is happening. So here we have a myosin, and it's double bound. So the myosin tail is wrapped around each other, so you have two heads. And the heads are what basically pull the filament. And in doing so, because it's attached to two of these, it can't really go anywhere. But by pulling them forward, basically what it does is it pulls the back end. So if I have something that I'm pulling forward, it's going to end up going like this. And when it goes like this, watch the video for a second. All right, so it starts like this, but then it starts to pull them. And so it's going to go like that. And if this part here, my hands are attached to the back, by doing this, I'm pulling the back forward. So by having the myosin here, it's pulling the back this way, and it's going to basically cause this part of the cell to contract forward. And so that's why we see a lot of the myosin at the trailing edge. So the leading edge has ARP2-3, nucleating the branch treadmilling, cofillin right behind it to depolymerize the minus ends to promote treadmilling as well, and then myosin at the back end pulling these longer actin filaments forward, basically by walking these up.
The shape of the cell is highly polarized. A broad, thin veil of cytoplasm called the lamellipod is at the front of the cell, and the back of the cell is tapered into a thin tail. The nucleus and major organelles are located in the cell body. A network of actin filaments fills the entire cell, while contractile myosin molecules are located in the cell body. Cell crawling takes place in 4 stages. In the extension stage, the cell pushes the lamellipod forward. This movement is driven by rapid polymerization of actin filaments at the leading edge of the cell membrane. In the next stage, the lamellipod adheres to the underlying surface using specialized membrane adhesion sites. Firmly anchored, the cell is now ready to generate the force to move the cell body forward. The third stage is translocation of the cell body. This is accomplished by contraction of actin and myosin bundles at the lamellipod cell body border. After the cell body translocates, the back of the cell releases from adhesion sites and moves forward.
All right, so in summary, hopefully you can see how the coordination allows the cell to move. We have branched polymerization at the lamellipodia leading edge for protrusion. Then we're going to have adhesion molecules, which we'll talk about later in this unit, to attach down so that you have force, right? So a car can spin its wheels, have a lot of movement, but actually not pull anything forward unless it has traction. So that adhesion allows the cell to have traction. And then we have the myosin working kind of towards the back end, pulling it forward. All right, so this actually can be coordinated by cell signaling. So that makes sense because you want the cell to be reacting to chemoattractants, or you want the cell to be reacting to signals from the outside of the cell that basically tell it where to go, what to do, and even if it's supposed to be moving at all. So here we have a quiescent cell. That means a cell that's not moving. And so this quiescent cell, you can see the actin, and so you have a lot of actin standing around the periphery, but you also have a lot of kind of stress fibers. It's basically staying put. And so the rest of these other 3 cells, B, C, and D, have activation of certain signaling molecules that can control the type of actin filaments that are made. All right, and so this would be a little bit abnormal because these cells have that activation everywhere. Whereas most cells moving, you would want to activate that on one side so that that side is moving forward and the other side moving backwards. So don't let that confuse you. I'll talk about that again in a second. But this RAC activation shows RAC being activated around the whole cell. This CDC42 activation shows CDC42 being activated around the whole cell, and Rho activation around the whole cell. Normally these would be coordinated, so one side has something going on to help it move forward, and the other side has something going on to move backwards. But what I want you to look at is, based on what you know, what do you think these cell signaling molecules activate? All right, what part or what format of cytoskeleton actin filaments do they regulate? So see if you can figure that out just kind of based on looking at this and answer the Edpuzzle question there. All right, so we can see that when CDC42 is activated, we get those long, thin protrusions, and we know these are filopodia. So CDC42, basically activation causes filopodia to form. And we can imagine that CDC42 activation is a signaling molecule that basically ends up resulting in fibrin being active. And so remember, fibrin is what bundles those actin filaments together, and also would result in formin being active. And formin is that hula hoop hat that basically with the 2 arms that's pulling and bringing the actin together. So if the right signal on the outside ends up activating CDC42 on the inside, you'll get filopodia. On the other hand, if you see RAC here, now we have branched treadmilling. And it's weird because the whole cell is trying to go in all directions at once. So if this is a real cell, it really has no directionality. It's just trying to move, and it would kind of have its own tug of war. Same with this one here. But this shows us that RAC is basically going to activate lamellipodia. So you can imagine that RAC activation is basically activating the ARP23 complex at the leading edge and cofillin right behind it for branch treadmilling. And then finally we have Rho activation, which really is the stress fibers. So this is where attachment would happen and depolymerization or pulling forward. And so again, normally we would have the Filopodia in the rack, the CDC42 activation of Filopodia, the rack activation of Lamellipodia at one end, and the stress fibers kind of towards the middle and the other end if the cell was moving. So that coordination really is what can cause this. So again, rack is going to lead to some signaling activation, and it activates ARP, like I said, and it activates filamin, all right, for the cross-linker, and it can even activate the cofilin behind it, things like that. So this is going to give you the branched actin, Lamellipodia, sorry. Rho, on the other hand, activates the formins and basically is going to cause stress fibers. Right? So Rho activates stress fibers, RAC activates Lamellipodia, CDC42 activates Filopodia. Don't forget that. So let's go all the way full circle now to our neutrophil. That neutrophil was following our bacteria, and the bacteria has lots of membrane proteins and especially lots of sugars that basically accidentally get broken off, sloughed off as it's moving. So there's a high concentration of these bacteria proteins and sugars near the bacteria. And so as this bacteria moves, these are what are the chemoattractants. So the neutrophil has receptors on all sides. But think back to Unit 4. We know that we have threshold. So which of these receptors are going to hit its threshold and bind the chemoattractants first? Well, it happens to be the side that's closest to this. So if the bacteria moved and was over here, well, now these receptors will hit threshold first, and allowing the neutrophil to follow it. If the bacteria ended up moving over here, these will hit threshold first. So basically you have receptors on the leading edge, the edge towards where it should go, following the bacteria, that hit threshold, and the other ones do not. So if this is the side we want it moving forward, what signaling molecule should we activate over here? Well, the signaling molecule that should be activated over here is the RAC for actin polymerization, and the signaling molecule that should be activated behind it is Rho. So the ligand binding receptor activates RAC, and then where there is no activation, Rho is of the receptors, Rho is activated by default, and so now we have one end. If we go back here, now we have one end that has Lamellipodia moving forward, and the back end that has Rho, and it's all based on which receptors hit threshold first. So the Lamellipodia will become activated here, stress fibers and retraction activated here, and that allows the bacteria, or the neutrophil to follow the bacteria no matter where it goes, because this can shift. If suddenly these receptors hit their threshold first, well, then this is going to activate RAC over here for Lamellipodia now here, and Rho will now be activated over here, and basically the cell will turn like we saw in that video. Alright, so in summary here, hopefully we've brought it full circle, how a chemoattractant can organize the cell's cytoskeleton in such a manner that it moves in the right direction. So, at the front edge here, we are going to activate ARP, and that's going to nucleate these branched actin filaments towards this direction, and cofilin right behind it is going to depolymerize them. Now we get branched treadmilling in the right direction. And behind it, we are not only going to activate kind of the actin stress fibers, but we also activate myosin to pull this back end forward. Now we have a polarized cell moving in unison. So it's not trying to move just randomly, it's moving in this direction with the leading edge coordinated with the trailing edge. And again, that can change, allowing this neutrophil to move and basically chase the bacteria. And here that is again, just for your viewing pleasure.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells.
We've got RAC going to be activated over here because that's near the bacteria. And we'll have Rho being activated back here for myosin pulling it forward.
Release a chemoattractant
Branch treadmilling here. Contraction in the back.
The neutrophil becomes polarized and starts chasing the bacteria.
Retraction.
The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.